Hey guys, welcome to my channel Research Updates. As researchers have learned more and more about cancer immune system interplay, many anti-tumor immunotherapies have received FDA approval and are currently being used to treat a variety of cancer types. However, despite these developments, a lot of information regarding immunity in general and how the immune system fight cancer remains unclear. Now, researchers from Harvard Medical School are deploying CRISPR based gene editing in a new way to study the function of immune genes. CRISPR based gene editing is a tool developed just over a decade ago, has become a standard technique of biological discovery, providing relatively quick insight into the function of individual genes and target for new therapies. Programmable CRISPR based gene editing becomes such a powerful tool for biological research that its discoverers also won the Nobel Prize in Chemistry in 2020. The CRISPR gene editing system uses an enzyme called Cas9 that acts like a pair of molecular scissors that cuts both strands of DNA and in doing so disrupt or knock out the function of a gene. This approach uses a complementary piece of RNA that matches the gene and serves as a guide to determine which gene to knock off. It's a very adaptable method for rapidly knocking out and examining the function of almost any desired gene. However, this method is not without difficulties. The primary obstacle is that it is challenging to modify immune cells without altering their biology. And also, immune cells interact with many other cell types inside the body, which makes it difficult to examine immune cell behavior in all of its complexity within a living organism. But the researchers prefer immune studies to take place inside a living organism such as a mouse. This is because immunological studies inside living organisms are far more reliable at capturing the complexity of cell-to-cell interactions as they occur in the body than in petri plates. Since it is difficult to use CRISPR editing inside the body, immune cells usually need to be isolated and manipulated in petri plates using this technique, and then edited cells are put back into the body. Also, immune cells have only been treated with CRISPR to silence a single gene at a time. But since our cells have hundreds of genes, what if we wanted to silence numerous genes in various cell types at different times in the same animal? With CRISPR, researchers used to replace the mice' native bone marrow stem cells with these after using CRISPR to knock out the genes of interest. This system is referred to as chimeric immune editing or CHIME. Their goal was to see whether they could improve CHIME to make it more accurate and adaptable. Using CRISPR, they disrupted genes in modified cells after they were inserted into animal, also used it to knock out two genes at once in multiple cell types and knock out two different genes at different times. The ultimate goal behind this research is to better understand the immune system, particularly in its capacity to fight cancer. Moreover, the benefits could extend beyond cancer and can be applied to many other diseases driven by the immune system. Thanks for watching.